In this video, we are going to talk about coronavirus testing and its implications. It is based on an article published in Nature. According to the World Health Organization, coronavirus testing was declared as the top priority. Singapore and South Korea responded, USA not. Let's see what happened. The article in Nature quotes that every single time we have an outbreak, we are one step behind in that we don't have a rapid diagnostic to detect that new organism, says Charles Chu, an infectious diseases physician at the University of California, San Francisco, who is working with Mammoth Biosciences. Here I will share with you information regarding current testing available for COVID-19. Nose and throat swab is taken from the suspected patient. Viral genetic material is isolated from the patient sample. COVID-19 causing virus genetic material is in the form of ribonucleic acid. The ribonucleic acid of virus genetic material is converted to DNA and then polymerase chain reaction is performed, and the technique involved is known as reverse transcription polymerase chain reaction. The outcome of PCR test for COVID-19. Virus genetic material is amplified and detected. This technique only works if the patient has active viral replication. A person who has recovered from virus cannot provide any information about his or her disease status. Several countries have customized primers, which means that they are synthesized indigenously, and different types of PCR are being performed. However, there is one potential problem with the PCR test, that is false positive, which is particularly when manpower conducting the test has not been trained properly. There were some countries who listened to the advice by WHO and some who didn't. One of the quick responders is Singapore, where the virus is almost under control. Singapore developed a diagnostic test for the SARS coronavirus 2 at the emergence of virus in China. Logistics were put in place, including developing laboratories' capacity for testing of coronavirus with implementing appropriate biosafety measures, laboratory personnel trained in implementing biosafety while working with the infectious samples, reagents and tools, polymerase chain reaction setups, etc. Among the slow responders, US is the one. In a congressional hearing on 12 March 2020, National Institute of Allergy and Infectious Diseases Director Anthony Fauci said that the country was failing in its ability to test people and explained that the United States was not set up to offer tests at the pace currently needed. However, the heroic efforts of a university in USA are worth mentioning. As the federal response lagged, virology research labs at the University of Washington in Seattle emerged as the first major US testing operation, where researchers adapted the WHO protocol to work with reagents and platforms they had to hand. Dozens of other labs and research consortia have now followed suit. Ideally, what is needed for COVID-19 testing is serological test. These are the tests that look for antibodies in the human blood or bodily fluids. Antibodies are produced as a result of antigen or foreign material entering into the body. Human body produce antibodies circulating in the blood for viral infectious disease like COVID-19. Benefits of serological test. We can get information if someone has been infected with the viral infection in the past, as that person will be having antibodies for COVID-19. Asymptomatic individuals can be quickly screened for COVID-19 disease, as asymptomatic individuals keep spreading virus in the community. Viral spread can be estimated in a community relatively faster with lesser chances of false positive. Efforts are ongoing to develop serological test. Like here is an article that mentions about a serological assay to detect SARS coronavirus 2 seroconversion in humans. This article is a preprint and has not been peer-reviewed. Scientists are also working on innovative technologies for COVID-19 causing virus detection. Sherlock test, University of Washington School of Medicine, detector test, Mammoth Biosciences, a biotechnology company in South San Francisco, California. Sherlock and detector use the CRISPR machinery's ability to recognize specific genetic sequences and cut them, followed by its amplification or detection.